Good afternoon, everyone. Today, my talk will be on best practices for regulatory submissions that include physiologically-based pharmacokinetic modeling and simulation approaches that are specific to locally acting generic drug products. Bioequivalence for locally acting drug products have a unique aspect, and I will highlight that throughout the talk. But I will mostly focus on physiologically-based pharmacokinetic modeling and specifically what are the best practices for development of these models, assessing their performance, how they can be used to perform virtual bioequivalence assessments, studies and assessments, and how they need to be reported and documented in regulatory submissions. And I will highlight the most important aspects of these best practices with an example, an approved ANDA for a complex topical drug product. So locally acting drug products are meant to be applied to the eye, the nose, the lung, the skin, and administered to act locally in the gut. They aim to act locally at the site of application, but they may also act systemically, such as, for example, transdermal delivery systems. And you can appreciate from these figures the complex physiology and anatomy of the target sites for all of these drug products. For the non-modelers in the room, I will just go over very quickly what PBPK modeling is. It's a quantitative modeling and simulation tool where every organ in our body is represented by a compartment, can be represented by a compartment, and the drug that gets in or gets out is described by a differential equation. The dimensions of these different compartments, these different organs, are informed by physiological values, the blood flow to our liver, or the mass or the volume of our heart, for instance. The strength with these models, this modeling approach, is that it can provide us with the link with between what we can measure, which will be formulation attributes or systemic drug exposure, with what we would like to know, and that would be local drug concentrations. And this is key when we are trying to establish bioequivalence for these locally acting products. PBPK modeling was introduced in the 1960s or 70s. However, it has gained a lot of traction, especially with regulatory submissions the last 20 years. And this is a list of how it can be used in the realm of the new drug case and the new drug agents. But in this talk, I will be focusing in this workshop, we are focusing on generic drug products. How PBPK modeling can be used for the development of product-specific guidances, how it can support alternative B approaches for demonstrating bioequivalence, and how it can be used to extrapolate B assessments from healthy to other subpopulations, disease, pediatrics, elderly, and how it can allow us to define a safe space for drug product specifications. Dissolution specifications may be an example under this category. If you go over our PSGs for complex locally acting drug products, you will see a general trend. We usually the agency provides an option when the reference and the test drug product are very similar, Q1, Q2 the same, where in this case we would ask for in vitro characterization and Q3 similarity for these products. And we can supplement our recommendations with in vivo studies with clinical endpoints or PK endpoints. Under this scenario, PBPK modeling can be used to support drug product development under the Q3 characterization, or it can be used to support alternative B approaches that do not include in vivo studies. The second scenario that is entertained in our PSGs is when the reference and the test drug product are not the same. They're not Q1, Q2 the same. And in this case, we usually ask for a clinical endpoint study. And again, in this case, under this scenario, we could use PBPK modeling to support alternative B approaches. However, this is a scenario and approach that is still a work in progress. So the ultimate goal when we're using PBPK modeling is to predict exposure drug concentrations either locally or in our blood, the systemic circulation. And I will go over with you the workflow that we follow when we develop these models. First, it will be a quick overview, and then we will break down every single step. So the first step is to develop the model. We decide model structure, and we incorporate drug product attributes, physicochemical properties of our API. In vivo and ex vivo data can be used to inform model parameters and the physiology, whether the drug will be simulated in a healthy or a diseased population. The assessment of the model performance is a key part, and it can be done by verification or validation, and I will go over these two terms in detail in the upcoming slides. And at some point, we might need to refine and optimize some of our model parameters. So this is part of the model development and refinement process. So the model development process begins with an internal reporting and documentation. We need to have a modeling analysis plan that outlines the objectives, what are the model inputs, what are the reference sources for these model inputs, and this is a living document. It changes throughout the process. It is, however, the backbone for a model analysis report that is a far more technical document, and it is the basis for the conclusions that we will come up with that will eventually inform decisions. So here's how we go about develop a model. We will need model input data on the drug substance, our drug product, and our physiological system. This input data will need to be of good quality, low uncertainty. They need to be experimentally derived, and they need to be biologically plausible. The structure of the model needs to be justified and supported by data or sensitivity analysis. We need to take into account parameter and population variability, as well as correlations between the parameters that we are using in our model. And finally, we need to be able to describe a physiology of the disease or the healthy population as accurately as possible into the model. And after this process is complete, we will need to move on to model verification. And this is essentially a test, a check of how differential equations and code describe the different model components accurately, and whether the engine that we are running the model is performing as expected. A very important part of model verification is biological plausibility. We need to check whether the physiology, the route of administration or the site of action, the drug substance ADME properties, and the drug product attributes are accurately represented into the model. Overall, model performance assessment is a process that aims at demonstrating that the proposed PBPK model is appropriate for the modeling purpose or question asked for the particular drug product and the study population, and is robust enough to respond to perturbations in uncertain parameters. This is a long and intense, loaded definition, but we will go over it in more detail. First, when we are assessing the performance of the model, we are performing what we call model validation for the intended purpose of the drug product of interest. And this is essentially a comparison between observed data that you see on the left as symbols and their error bars versus what the model predicts, which is the red line. The observed data that we're using for model validation needs to be of good quality. They need to represent clinical scenarios that are relevant, and they need to be drug product specific, and they need to capture the variability of the drug product and the population. When it comes to the acceptance criteria, these reflect the regulatory impact of the decision that is associated with the model. However, they need to be established a priori and used consistently. A very important part of model performance assessment, which is unique for locally acting drug products, is the platform performance assessment. It is key for locally acting drug products because For these products, we don't have local concentrations that would allow us to assess the performance of the model locally. Therefore, our confidence into the model increases when we see a platform performing, the platform that is used for the model performing adequately. The platform performance assessment involves developing models under the same principles as the drug product of interest. These models are for APIs of similar physicochemical properties and ADME characteristics. The dosage forms that will be modeled are of variable compositional complexity for the same route of administration, and these models are not used for model development. If the model is intended to be used to a disease population or a special population, then one will want to demonstrate through the platform assessment performance that the interplay between the formulation and the population or the physiology is well captured. PBPK models for ocular, inhalation, nasal, and dermal products, they are validated using a variety of data. There's consistency in what data we would like to use in vitro or ex vivo, as well as in vivo on the systemic and local exposure in humans. However, what is slightly different is in the case of the ocular PBPK models, we may need to validate the model using animal data and then extrapolate to humans. There are a number of considerations and challenges associated with this task. The first one is data availability for model validation, for establishing in vitro and in vivo correlations, relationships, and for describing that interplay between our product and the physiology. We need to keep in mind that systemic data do not always reflect local concentrations, and that the model is validated only for the range of conditions that are covered by the data set that we used for the validation. And in some cases, even after this rigorous process is complete, we might not have a good agreement between observations and predictions. So in this case, we will have to follow what we call a middle-out modeling approach, which involves parameter optimization. And this is obviously indicative of a knowledge gap. As we go about optimizing parameters into our model, we need to keep in mind that we have been working on the development of a fit-for-purpose model, that we might have to encounter issues of identifiability, structural and numerical identifiability. There might be parameter collinearities that we need to take into consideration. Our output needs to be biologically plausible, and we might have to undergo, our model might have to undergo additional verification. And after we have this model that behaves, that performs adequately, we can use it to perform virtual bioequivalence assessments. We can generate population predictions for the reference and the test drug product and perform statistical analysis. Or we can use it to identify clinically relevant drug product attributes by performing sensitivity analysis. Again, capturing the variability of a population or of a drug product is key here, because that would allow us to generate realistic population predictions. And our sample size may be also another consideration, since we need to keep in mind the potentially highly variable pharmacokinetic behavior of a product of an API locally. So I'm sure that this figure makes more sense to you now. It describes, it outlines the workflow towards the development of a PBPK model, one for the reference and one for the test drug product. We get our predictions. These are the red and the blue line on the right. And these predictions allows us to determine, to answer the question, is the reference and the test product, are they bioequivalent? So the next question answering your mind is, how do we communicate, how do we share this task with the regulatory agencies? There are two guidances, one from the FDA on the left and one from the EMA on the right, that you can use to guide you through that. They're very useful. However, I would like to highlight a couple of points First, how do you construct your modeling? How do you put together your modeling and simulation report? Key aspects of key points that need to be covered in this report is the model development and model assessment performance. What are the model assumptions, the model limitations, and how they're taken into consideration when you're performing your simulations, and whether your conclusions are data and science driven. We expect that executable files, input and output files are shared with the agency. And if there's amendments or discussion going on during the review process that results in updated materials, we would appreciate if these materials are submitted to the agency. So our take-home messages. PBPK models for locally acting products can be used to support development of drug products prior to approval, as well as support alternative B approaches. Model development is an intense and research-demanding process because of the complexity of the products and because of the complexity of the models. And there are limitations on the data availability in model development and validation, and this is another challenge associated with the development of PBPK models for locally acting drug products. That is why we encourage applicants to engage with the agency early in their drug development program through the pre-NDA meeting request program under GDUFA 2. And to highlight certain aspects of the best practices that I went over, and to also demonstrate that this can actually be done, I will go over a dermal PBPK model that was submitted to the agency supporting an approved ANDA, ANDA 211253. This was for a generic for diclofenac sodium topical gel 1%. The generic was very similar to the reference product. It was Q1, Q2 the same, and Q3 similar. And the applicant submitted the dermal PBPK model instead of conducting the PSG recommended comparative clinical endpoint study. They used a commercially available software platform to develop their dermal PBPK model, and they incorporated physical chemical properties of diclofenac as well as its ADME characteristics, and they used the platform to develop two models, one for the reference and one for the test drug product. The two models were different as they were, they incorporated different formulation attributes. The applicant evaluated the assessment of the platform by developing more than 10 PBPK models for transdermal delivery systems and topical dermatological products. They simulated multiple doses, product strengths, and dosing regimens. And the performance of the models, of these more than 10 models, as well as the platform, was evaluated as satisfactory. And then they focused on the development of their dermal PBPK model for diclofenac. They used literature and application data, and again, they simulated a number of product strengths, dosing regimens, routes of administration, local and systemic exposure data. They developed one model for the reference and one model for their drug product, and you can see the two figures on the left. That the model described the data relatively well. The red dots are the observed data, and the blue line is the model prediction. Now, this model was intended to be replacing a clinical endpoint study. So for the agency, it was very critical that the performance of the model was adequate, not only describing systemic exposure, but also local drug concentrations. For that reason, we worked on further refining the model to improve the synovial fluid exposure. The synovial fluid is the presumed site of action for this drug. What we did is to include protein binding in all of the skin layers. We updated product attributes with internal data, and we optimized certain partition coefficients by leveraging observed local drug concentrations. And I would like to draw your attention to the lower graph here, where you can see that the black dot is the observed data. The blue line is the models, the applicant's model, and the red line is the agency's model. It was significantly, its performance was significantly improved. So we conducted virtual bioequivalence assessments using this refined model. You can see how the predictions look like in the systemic circulation on the left and in the synovial fluid on the right. The two products were found bioequivalent and the product was approved. Overall conclusions for today: PBPK models can be used to inform drug product development decisions and support alternative B approaches for generic locally acting drug products. The applicants are encouraged to follow best practices as these are outlined in guidances and other public forums such as the one here today. And we also encourage the applicants to engage with the agency early with us through the pre-NDA meeting request program and get feedback on their modeling approaches as well as the alternative B approaches. Acknowledgments, and I would also like to make you aware of our extensive research portfolio on modeling and simulation approaches for locally acting drug products. Thank you very, very much for your attention.